The kidneys have a lot more things they can do, but it takes them longer to do it, right? So there's basically two sides to the acid-base equation at any given time. There's what the lungs are doing, which is the now, the minutes, and then there's what the kidney is doing, which is the hours to days. Okay, so how do the kidneys participate in acid-base balance? Well, first off, you know, hydrogen, oh, this is bicarbonate. Bicarbonate, freely filtered, right? Yes, it's a negative molecule, but it's very, very small. So it does pass through the dense layer. Most of that bicarbonate, 85%, obligately reabsorbed. Not surprising, because bicarbonate is the way we use the lungs to get rid of acid. So we want to hold on to it. It's precious, right? So that's our obligate reabsorption. Early part of the distal convoluted tubule, we get a bunch more reabsorption. And then in the rest of the kidney, that's where we're going to have our regulatable, either reabsorption or secretion, depending on what the status of the body is, what the pH of the body is. So the way the kidney participates, number one, it secretes hydrogen ion. It reabsorbs filtered bicarb, okay? And then it produces new bicarbonate, which is some of this over here. And I'll show you the pictures of that in a minute. So because bicarbonate can be blown off, as I like to say, in other words, the CO2 can be removed and blown into the air and make the hydrogen ion into a water, right? Bicarbonate is held onto by the kidney because it's an effective base. It's a volatile base. So most reabsorption is obligate in the PCT, but there is regulatable absorption in the later nephron. Okay, so in the cells of the proximal convoluted tubule, the thick ascending limb, and the early DCT, all of these use this sodium hydrogen exchanger. Where are you? This guy, right? So sodium is falling down its concentration gradient, right, from high to low. Hydrogen is going up its, at least typically. Remember, urine pH can change pretty dramatically. So which direction is which can vary, but usually sodium's falling down, hydrogen ion is going up. So it's a countertransporter. Now, here's where it gets, all of these things have multiple reactions to them because hydrogen ions are so reactive, right? So in the tubular lumen, we have sodium and bicarbonate. Okay, we excrete a hydrogen ion. Woohoo. Okay, so we got rid of a hydrogen ion from the body. It's going to combine with the bicarb to make carbonic acid. And then it's going to split carbonic anhydrase into CO2 and water. Now this is where it gets weird. CO2 diffuses readily across membranes. Remember, it's lipid soluble, doesn't need a transporter. So CO2 is going to dissolve into the tubular cell. Inside the tubular cell, there's going to be plenty of water, right? So we're going to combine this again. Oh, look, carbonic acid, or carbonic anhydrase again. It's going to split apart into hydrogen ion and bicarbonate. The bicarbonate will be reabsorbed, and one might say the hydrogen ion has come out here, right? So I mean, wherever the hydrogen ion might have come from there, might have come from somewhere else. Then the bicarbonate is secreted, so in other words, it's reabsorbed into the tubular fluid, right? Because this is peritubular capillaries. Bicarb back into the tubular capillaries. And then off to the lungs, where it can be used to breathe off. We get rid of a CO2 and consume a second hydrogen ion, right? So essentially, we are clearing one hydrogen ion here, and then another hydrogen ion at the lungs from here. So we're getting rid of two hydrogen ions by reabsorbing one molecule of bicarbonate. So it's very useful. 95% of the bicarb is reabsorbed in this way. Note the bicarb itself doesn't ever cross the membrane on this side, right? It enters as bicarb, it leaves as water. You see, the bicarb never comes in, but it is remade inside, and that bicarbonate that's used, that is made inside the cell, then can be used later on. So this is regulatable. If hydrogen ion is high in the body, we'll make more bicarbonate, and vice versa. Right. All right, your intercalated cells are the ones that can flip their poles, right? They can face acid way or they can face base way. We find them in the late distal tubule and the collecting ducts. Our major player here, the hydrogen pump, so proton pump, pumping hydrogen ions into the lumen. In other words, secreting acid, secreting hydrogen ions into the urine where it can be removed. So we have ATPase, a hydrogen potassium exchanger, okay? Hydrogen is going up its concentration gradient. Potassium is also going up its concentration gradient, right? So that's a busy transporter because we're pumping two molecules both up their concentration gradients, but that is what's happening. And then we have a bicarb chloride exchanger on the basolateral membrane, on the tubular capillary, on the peritubular capillary membrane, right? All right, so hydrogen ion gets pumped out. Where does the hydrogen ion come from? Well, it may have come from CO2. Okay, CO2 is ubiquitous, right? So CO2 combines with water, carbonic acid, bicarbon, hydrogen ion goes towards the tubule. Bicarbonate is then reabsorbed. That's a pattern we just saw. That bicarbonate that's reabsorbed goes to the lungs. It's used to pull off another hydrogen ion by breathing out a CO2. Okay, and then the potassium is just first moving up its concentration gradient and then down its concentration gradient. All right, so these are the type A cells. The type B cells, same system, flip the membranes. All right, so in other words, these transporters are on this side and these transporters are on this side in the B. And I've got a picture of B cells in one of the other kidney talks, so you can go and look back at that if you need to. Okay, so that is what the cells are doing. We also have some reactivity that's happening in the filtrate itself because of buffering characteristics. Okay, so if we're over here, this is what's coming out of the Bowman's capsule and the nephron, right? So we're going to have a bunch of sodium. We're going to have some sodium phosphate. Why? Because it was filtered. It's a small molecule, freely filtered, right? So it's going to end up in the tubular lumen. Okay, now what's interesting is this NaHPO4, this, remember, phosphate buffers have multiple states or stages. So we can take our hydrogen ion that we've excreted and we can add it to the NHPO4 and get NH2PO4. Now the beauty of this is this hydrogen ion, if it wasn't for the buffer, this hydrogen ion would tend to decrease the pH. In other words, elevate the concentration, right? Well, every time you do that, you make it a little harder for this pump, for this countertransporter to do its thing. In other words, if you have lots and lots of hydrogen ions in the tubular lumen, this is not going to be able to pump more out. The gradient's too steep. But we have buffers that absorb it. So this hydrogen ion isn't going to change the pH. It's just going to get nabbed by the sodium hydrogen phosphate group, right? So we can excrete a hydrogen ion without a change in pH. So hydrogen ion out, and that hydrogen ion secretion nets us a bicarbonate reabsorption as well, right? Because the CO2 and water went in, hydrogen ion got excreted, bicarbonate got reabsorbed, that goes to the lungs, we get rid of another one. See a pattern? We're going to see this kind of thing a lot. Every bicarb is a way to get rid of a hydrogen ion, and that's kind of the way the body looks at it. Okay, so that's in the, so this is a buffer system, right? This is the, the sodium phosphate buffer system in the tubular lumen. Interestingly, that same buffer system, it does the same thing inside of cells too, same process. But here we're using it to kind of, we're using it to get rid of more acid without a change in urine pH. Because for every bit of urine pH change, it's harder to put more hydrogen ion in there. All right, we also have ammonia. Ammonia is another buffer system that plays a role particularly in the tubular lumen. And as your book says, more important quantitatively than the phosphate buffer. In other words, this involves more molecules, more units of acidity than the phosphate buffer does. All right, so it all starts with glutamine. Glutamine is an amino acid. It's fairly ubiquitous, right? It is freely filtered and heavily reabsorbed, as all amino acids are, right? But nonetheless, some glutamine still does arrive in the tubular lumen. Now, it is brought into cells. There's a transporter for that. No need to know what that is, but remember, amino acids all have transporters. So this is going to come in. Now, inside the cell, we get this kind of cool reaction where glutamine is broken down into 2 bicarbonates and 2 NH4s. Now, the usefulness of this is, well, we got 2 bicarbonates out of it, right? We like those, send those to the lungs to do some things. But then the NH4, we can shuttle that out into the tubular lumen, and that NH4 is an effective way of getting rid of hydrogen ions as well, right? So we have our glutamine ammonia buffer system, right? So we get 2 NH4s that are secreted in exchange for sodium. So that's a good deal. We get sodium, we export NH4, and then the 2 bicarbonates are reabsorbed and moved to the lung. So this is yet another way of creating bicarb. Oh, proximal convoluted tubule. In the collecting system then, okay, the NH4 would still be in here. But here in the collecting system, our NH3 is going to diffuse out. We still have our proton pump pumping out. We can combine an NH3 and a hydrogen ion to make an NH4, and that's making a buffer again, right? So that is resisting the change in pH that would have made this pump work less well. So the NH3 becomes the base, right, where it's accepting the hydrogen ion that originally came from CO2 and water and carbonic anhydrase. So this is yet another example of create a hydrogen ion by breakdown of carbonic acid, reabsorb the bicarb, and then excrete the hydrogen ion, but don't change the pH because that makes it harder to excrete another one.